Welcome to this lecture on evolutionary biology. Today, we are going to explore a fascinating and fundamental topic known as isolating mechanisms. Have you ever wondered why, with so many different animals and plants sharing the same habitats, they do not all just blend into one giant uniform species? Nature has invisible boundaries that protect the unique genetic identity of every species. By the end of this session, you will understand exactly how these biological walls work, why they are absolutely essential for the diversity of life on Earth, and how we can even measure their strength mathematically. Let us dive in. To start our journey, let us think about something we use every day. Imagine two different smartphone operating systems, like iOS and Android. They both do similar things, but you cannot run an iOS application on an Android phone because their internal codes simply do not understand each other. In the natural world, animals and plants have their own biological operating systems. These take the form of specific genetics, physical traits, and mating behaviors. If a bird from one group tries to mate with a bird from a completely different group, their genetic codes might clash, or they might not even recognize each other as potential mates. This incompatibility is the first step in keeping populations distinct. Now that we understand the operating system analogy, let us look at the formal biological definition. In evolutionary biology, this invisible wall is called an isolating mechanism, or a reproductive isolating barrier. These are specific biological traits, behaviors, or environmental factors that actively prevent individuals of different species from successfully interbreeding. Why does this matter? Because these mechanisms ensure that even if two different species live in the exact same geographical area, like two types of frogs in the same pond, they will not merge into a single hybrid population. It is nature's way of keeping different groups separate, allowing them to evolve on their own unique paths without constantly blending back together. Building on that definition, scientists divide these isolating mechanisms into two broad categories based on exactly when they act during the reproductive process. The first category is prezygotic mechanisms. As the name suggests, these act before a fertilized egg, or zygote, can even form. They stop mating or fertilization from happening in the first place. But what if this first line of defense fails? That is where postzygotic mechanisms come in. These act after a zygote has successfully formed. Even if two species manage to mate, postzygotic barriers ensure the resulting hybrid offspring either die young or are completely sterile, meaning they cannot reproduce themselves. Understanding these two types of barriers leads us to a bigger question. Why do they matter so much in the grand scheme of life? Without these crucial barriers, the incredible diversity of life we see around us would simply not exist. Any new advantageous trait would quickly be diluted by constant interbreeding with other groups. Isolating mechanisms are the fundamental drivers of speciation, the process by which new species arise. Furthermore, in practical fields like agriculture and conservation biology, understanding these mechanisms is absolutely crucial. It helps us manage endangered species and prevents genetically modified crops from accidentally cross-pollinating with wild plants, which could wash away unique genetic adaptations. Now that we have established the conceptual importance of these barriers, let us look at how they work mathematically. We can represent the breakdown of isolation using the equation for the change in allele frequency due to gene flow. The formula is delta P equals m times the quantity P sub m minus P. Here, delta P represents the change in the frequency of a specific gene variant, or allele, in the receiving population over one generation. The symbol M is the migration rate, representing the proportion of the population that consists of new migrants. The variable P sub M is the allele frequency in the migrant population, and P is the allele frequency in the original resident population. When isolating mechanisms are perfectly strong, the migration rate M becomes exactly 0, meaning the populations are completely isolated, and delta P is 0. Let us apply this formula to a concrete example to see it in action. Imagine two populations of frogs. Before any barrier existed, the migration rate between the two groups was 0.20. The allele frequency for green skin in the migrant population is 0.80, and in the resident population, it is 0.30. We want to find the baseline rate of genetic change before any barrier existed. We substitute our given values into the gene flow equation. We subtract 0.30 from 0.80, giving us 0.50. Multiplying this by the migration rate of 0.20 gives us a final change in allele frequency of 0.10. This means the resident population's genetics were changing by a massive 10% every single generation. Now, let us see what happens when an isolating mechanism is introduced. A fast-flowing river forms between the frogs, acting as a strong geographic pre-zygotic barrier. This reduces the migration rate down to 0.05. The starting genetics have not changed yet, so our allele frequencies remain the same. We substitute the new migration rate of 0.05 into our equation, multiplying it by the same difference of 0.50. This gives us a new change in allele frequency of 0.025. Physically, this means the isolating mechanism has drastically reduced the genetic blending from 10% down to just 2.5% per generation. Over many generations, this massive reduction protects the resident population's genetic makeup, allowing them to evolve independently. We have just seen how a pre-zygotic barrier, like a river, stops gene flow. But what happens if those initial barriers fail? Sometimes, individuals from two different species do manage to mate and produce a hybrid offspring. When this happens, the second line of defense must kick in. Post-zygotic barriers ensure that these hybrids do not become a bridge for genes to flow between the two parent species. For the species to remain distinct, the hybrid offspring must be evolutionarily unsuccessful. They might be weak, unable to survive to adulthood, or completely sterile. This severe disadvantage is what ultimately maintains the invisible biological wall between the species. To understand exactly how strong a post-zygotic barrier is, we can calculate it mathematically. We measure the evolutionary success of these hybrids using a fitness value, denoted by the letter W. A fitness of 1 means perfect health and reproduction, while a fitness of 0 means complete sterility or death. The formula for hybrid fitness is W sub H equals V times F. Here, the symbol V represents the viability, which is the survival rate, or the chance the hybrid lives to adulthood. The symbol F represents the fecundity, which is the fertility rate, or the ability to produce offspring, compared to a normal individual. By multiplying these two independent probabilities together, we find the overall evolutionary success of the hybrid organism. Let us put this fitness formula to the test with an example. Imagine two different species of fruit flies mate and produce a hybrid. We are given that the hybrid fly has a survival rate, or viability, of 0.40. We are also given that its fertility rate, or fecundity, is 0.10. we need to calculate the overall hybrid fitness to determine the strength of the post-zygotic isolation. We substitute 0.40 for V and 0.10 for F into our equation. Multiplying these together gives us a final hybrid fitness of 0.04. Physically, this means the hybrid offspring are 96% less successful at passing on genes than normal flies. Because they are evolutionary dead ends, the two parent species remain completely isolated. Having calculated the effects of both types of barriers, let us visualize how they work together as a system. Look at this flowchart. At the very top, we start with two different species. The first block they encounter is the pre-zygotic barriers, which include things like habitat or behavioral differences. This is the primary defense. If this barrier holds, the process stops here. But if it fails, as shown by the downward arrow, mating occurs and a zygote forms. Now the second block, the post-zygotic barriers, takes over. These include hybrid inviability or sterility. As you can see at the very bottom, the ultimate result of this sequential defense system is a giant red X, meaning absolutely no gene flow. The genetic identity of the species is successfully protected. We have covered a lot of ground today, so let us summarize the most important ideas. First, isolating mechanisms are the essential biological and geographical safeguards that protect the unique genetic identity of every species on Earth. Second, we learned that pre-zygotic mechanisms act as the first line of defense, stopping different species from mating or fertilizing eggs in the first place. Third, if those initial barriers fail, post-zygotic mechanisms step in to ensure that any resulting hybrid offspring are unfit, sterile, or unable to survive. Together, these powerful barriers completely stop gene flow. By doing so, they allow populations to diverge over time, ultimately driving the magnificent process of speciation that creates the biodiversity we see today. Great job following along with these complex evolutionary concepts.